Lecture 1. Definition of entomology and importance of insects. Introduction to phylum Arthropoda. Definition of entomology and importance of insects. The term entomology is derived from two Greek words. Entomon means an insect and logos means to study. The term insect is derived from the Latin word insectum, which means cut into. Insects came into Earth 480 million years ago. Man came to Mother Earth only 1 million years ago. Out of 17 lakh living species on Earth, 9.5 lakh species are insects, 2.5 lakh species are plants, and 0.45 lakh species alone are vertebrates. Insects are harmful to man as pests of cultivated crops, animals, stored products, carriers of human diseases, and pests of household and industrial articles. They are also helpful as producers of honey, lac, silk, dyes, etc., pollinators of crops, and as natural enemies of crop pests. They also serve as important link in the foodweb of biological cycle in ecosystem. Insects are grouped with other animals with similar characteristics in the phylum Arthropoda. Introduction to phylum Arthropoda. There are about 1 to 2 million species of animals in the world, and more than 70% of these are insects. These animals are assigned to 29 phyla. Of these, only 9 phyla are large, and their representatives are common and familiar to us. These are called the major phyla. The other phyla are small and their representatives are uncommon and of uncertain relationships. Position of insects in animal kingdom and their relationship with other arthropods. Insects are invertebrates grouped in the phylum Arthropoda, arthro joint, poda foot, and subphylum Uniramia. Characters of the phylum Arthropoda are 1. Segmented body. 2. Segments grouped into 2 or 3 regions, tagma, known as tagmosis. 3. Renewable chitinous exoskeleton. 4. Grow by molting. 5. Bilateral symmetry of body. 6. Body cavity filled with blood and called as hemocoel. 7. Tubular alimentary canal with mouth and anus at anterior and posterior ends. 8. Dorsal heart with valve-like ostia. 9. Dorsal brain with ventral nerve cord. 10. Striated muscles with dark and light bands. 11. No cilia, hair-like vibratal structure on the surface of the cell. 12. Paired, segmented appendages. Phylum Arthropoda is classified into 7 classes. 1. Onychophora, claw-bearing, e.g. Parapatus. 2. Crustacea, crustashell, e.g. prawn, crab, woodlouse. 3. Arachnida, arachne spider, e.g. scorpion, spider, tick, mite. 4. Chilopoda, chilo lip, poda appendage, e.g. centipedes. 5. Diplopoda, diplo 2, poda appendage, e.g. millipede. 6. Trilobita, an extinct group. 7. Hexapoda, hexa 6, poda legs or insecta, ininternal, sect, cut, e.g. insects. Major phyla of animals. SL. No. Phylum approximate known species. Examples. 1 Porifera 4,500 sponges. 2 Platyhelminths 12, 700 flatworms, planaria, tapeworm 3 Nematode 12,000 roundworms. 4 Mollusca 1, 000 snails, octopus, oysters 5 Annelida 7,000 earthworms, leech. 6 Arthropoda 1 million crabs, ticks, mites, shrimps, scorpions, spiders. And insects 7 Echinodermata 6,500 starfishes, sea urchins, sand $8 chordata 43,000 fishes, amphibians, reptiles, birds and mammals 9 Coelenterata 80,000 corals, jellyfish, Hydra Lecture 2. Introduction to class Insecta. Insect dominance. The relationship between insects and other major groups in the animal kingdom. Animal kingdom. Invertebrata vertebrata subkingdom. Animals without a backbone. Animals with a backbone. Fish, amphibians, reptiles, birds and mammals. Phylum Platyhelminths, flatworm. Nemathelminths, roundworm, mollusca, slugs, snails, arthropoda. Class Onychophora. Parapatus. Crustacea. Crabs. Shrimps, lobsters, sobugs, crayfish, myriapoda. Centipedes, millipedes, symphilids, arachnida. Spiders, scorpions, mites. Ticks, insecta. Insects. Character. Crus ta cea. Arachnida. Chil. Insect dominance. Insects are the most dominant species on the Earth as they originated on Earth 480 million years ago. Among 1.7 million living species, 0.95 million species are insects. Insect order number of species. Coleoptera, beetles and weevils 3, 50,000 Lepidoptera, butterflies and moths 1, 60,000 Hymenoptera, bees, wasps and ants 1, 20,000 Diptera, flies and mosquitoes 1, 20,000 Hemiptera, bugs 98,000 Orthoptera, grasshoppers, crickets and locusts 20,000. Measures of dominance. 1. More number of species. 9. 2. Large number of individuals in a single species, e.g. locust swarm comprising of 10 number of individuals, occupying large area. 3. Great variety of habitats. 4. Long geological history. Reasons for dominance. There are several structural, morphological and physiological factors responsible for insect dominance. They are 1. Capacity for flight. 2. More adaptability or universality. 3. Smaller size. Majority of insects are small in their size, conferring the following physiological and ecological advantages. 4. Presence of exoskeleton. Insect body is covered with an outer cuticle called exoskeleton, which is made up of a cuticular protein called chitin. This is light in weight and gives strength, rigidity and flexibility to the insect body. 5. Resistance to desiccation. Insects minimize the water loss from their body surface through prevention of water loss, wax layer of epicuticle, closable spiracles, eggshell, conservation of water capable of utilizing metabolic water, resorption of water from fecal matter, use less quantity of water to remove the nitrogenous waste. 6. Tracheal system of respiration. This ensures direct transfer of adequate oxygen to actively breathing tissues. Spiracles through their closing mechanism admit air and restrict water loss. 7. Higher reproductive potential. Reproductive potential of insect is high, e.g. Egg-laying capacity, fecundity, of queen termite is 6,000-7,000 eggs per day for 15 long years. Short development period, e.g., corn aphid produces 16 nymphs per female, which reaches the adulthood within 16 days. Presence of special types of reproduction other than oviparity and viviparity like polyembryony, parthenogenesis and poidogenesis. Lecture 3. Comparative account of insect external morphology. Head, antenna. Head. The head of an insect is composed of a series of segments, which are specialized for food gathering and manipulation, sensory perception, and neural integration. The head bears the eyes, compound eyes, and ocelli, antennae, and mouthparts. The anterior part of the head is the frons. The anterior area below the dorsum of the head, between and behind the eyes, is the vertex. The area below the compound eye, on the side of the head, is the genna. The lip-like sclerite is the clypeus. Based on the inclination of long axis of the head and orientation of mouth parts, there are 3 types of insects' heads. 1. Hypognathous, hypo below, gnathous jaw. This type is called orthopteroid type. The long axis of the head is vertical, it is at right angles to the long axis of the body. Mouth parts are ventrally placed and project downwards, e.g. grasshopper, cockroach. 2. Prognathous, pro in front, ganthous jaw. This type is also called coleopteroid type. The long axis of the head is horizontal. It is in line with the long axis of the body. Mouth parts are directed forward, e.g. ground beetle. 3. Opistognathos, opisthobenend, gnathos jaw. Head is deflexed, e.g. grasshopper. 1. Hypognathous, hypo below, gnathous jaw. This type is called orthopteroid type. The long axis of the head is vertical, it is at right angles to the long axis of the body. Mouth parts are ventrally placed and project downwards, e.g. grasshopper, cockroach. 2. Prognathous, pro in front, ganthous jaw. This type is also called coleopteroid type. The long axis of the head is horizontal. It is in line with the long axis of the body. Mouth parts are directed forward, e.g. ground beetle. 3. Opistognathos, opisthobenend, gnathos jaw. This is also called hemipteroid type or opisthorhynchus type. Head is deflexed. Mouth parts are directed backwards and held in between the forelegs, e.g. stink bug. Structure of insect antenna. Antennae are also called feelers. They are paired, highly mobile and segmented. Antennae are located between or behind the compound eyes. All insects except Protura have a pair of antennae. Antennae are well developed in adults and poorly developed in immature stages. The antenna is set in a socket of the cranium called antennal socket. The base of the antenna is connected to the edge of the socket by an articulatory membrane. This permits free movement of antennae. The basal segment is called scape. It is conspicuously larger than succeeding segments. The second antennal segment is called pedicle, WHCIH immediately follow the scape. A mass of sense cells called Johnston's organ is present in the pedicel, which is used as a chordotonal organ in some of the insects like mosquitoes. Both scape and pedicel are provided with intrinsic muscles. The remaining annuli or flagellomers are known as flagellum or clavola, which lack individual muscle. Surface of the flagellum is supplied with many sensory receptors that are innervated by the deuterocerebrum of brain. Flagellum may vary in size and form. Function. Antenna is useful to detect chemicals including food and pheromones, chemicals secreted into air by opposite sex. It perceives smell, humidity changes, variation in temperature, vibration, wind velocity and direction. Antenna is useful to perceive the forward environment and detect danger. It is useful for hearing in mosquitoes and communication in ants. Rarely it is also useful to clasp the mate, e.g. flee and grasp the prey. Antennae vary greatly among insects, but all follow a basic plan. Segments 1 and 2 are termed the scape and pedicel, respectively. The remaining antennal segments, flagellomers, are jointly called the flagellum. Antennae function almost exclusively in sensory perception. Some of the information that can be detected by insect antennae includes motion and orientation, odor, sound, humidity, and a variety of chemical cues. Some of the most common types of insect antennae with which you should be familiar are illustrated below. E.g., odonata, e.g. e.g., odonata, e.g., Coleoptera. e.g., Lepidoptera, e.g., Diptera. Types of antennae. 1. Cetaceous. Bristle-like, size of the segments decreases from base to apex. E.g., leafhopper, dragonfly, damselfly. 2. Filiform. 2. Filiform. Thread-like, segments are usually cylindrical. Thickness of 1. Cetaceous. Bristle-like, size of the segments decreases from base to apex. E.g., leafhopper, dragonfly, damselfly. 2. Filiform. Thread-like, segments are usually cylindrical. Thickness of segments remains same throughout. E.g., grasshopper. 3. Maniliform. Beaded segments are either globular or spherical with prominent constriction in between, e.g., termite. 4. Serrate. Saw-like segments have short triangular projections on one side. E.g., longicorn beetle. 5. Unapectinate. Cone-like segments with long slender processes on one side, e.g., sawfly. 6. Bipectinate. Double cone-like segments with long slender lateral processes on both the sides, e.g., silkworm moth. 7. Clavate. Clubbed antenna enlarges gradually towards the tip. E.g., blister beetle. 8. Capitate. Knobbed terminal segments become enlarged suddenly, e.g., butterfly. 9. Lamellate. Plate-like antennal tip is expanded laterally on one side to form flat plates, e.g., lamellicorn beetle. 10. Aristate. The terminal segment is enlarged. It bears a conspicuous dorsal bristle called arista, e.g., housefly. 11. Stellate. Terminal segment bear a style-like process, e.g., horsefly, robberfly. 12. Plumose. Feathery segments with long whorls of hairs, e.g., male mosquito. 13. Pelos. Hairy antenna is less feathery with few hairs at the junction of phlegolomers. E.g., female mosquito. 14. Geniculate. Elbowed scape is long remaining segments are small and are arranged at an angle to the first resembling an elbow joint. E.g., ant, weevil and honeybee. Lecture 4. Insect mouth part and types of mouth parts. Mouth parts. Just as insects take on many different forms, they also possess a variety of different mouth types, each of which can be grouped under one of two main categories: chewing, mandibulate, and sucking, haustellate. Mandibulate mouthparts, like the ones illustrated below, are believed to be the most primitive. All others, including those categorized as haustellate, are presumed to have evolved as modifications of this basic type. The 5 primary parts of the insect, mouth, are 1. The clypeus. 2. The upper lip, or labrum. 3. 2. Jaw-like structures, or mandibles. 4. The maxillae, sing. Maxilla. 5. The lower lip, or labium. The maxillae and labium are divided into various substructures, which include the galea, paraglossa, glossa, and the maxillary and labial palps. Haustellate mouthparts are primarily used for sucking up liquids, and can be broken down into 2 subgroups, those that possess stylets and those that do not. Stylets are needle-like projections used to penetrate plant and animal tissue. Examples of insects with stylets include hemiptera, true bugs, diptera, flies, and siphonoptera, fleas. Some haustellate mouthparts lack stylets. Unable to pierce tissues, these insects must rely on easily accessible food sources such as nectar at the base of a flower. One example of non-stellate mouthparts is the long siphoning proboscis of butterflies and moths, order Lepidoptera. Although the method of liquid transport differs from that of the butterfly's proboscis, the rasping sucking rostrum of some flies is also considered to be haustellate without stylets. Adaptations of the mouthparts. Fundamentals of entomology. 5. Fundamentals of entomology. Adaptations of the mouthparts. Insect mouthparts. Top left, chewing, top right, sponging, center, piercing sucking, bottom, siphoning. F. W. Zettler, Cornell. Types of insect mouthparts. Mouthparts of insects vary to a great extent among insects of different groups depending upon their feeding habits. They are mainly of two types, viz. Mandibulate feeding mainly on solid food, and haustellate feeding mainly on liquid food. 1. Biting and chewing type, e.g. cockroach and grasshopper. It is the primitive type of mouthpart and consists of the following parts. I. Labrum. Upper lip. It is flap-like, bilobed and attached to the clypeus by an articular membrane. It is movable. It covers the mouth cavity from above. It helps to pull the food into the mouth. It holds the food in position so that mandibles can act on it. It forms the roof of the preoral food cavity. E. Labrum epipharynx. Inner surface of the labrum is referred to as epipharynx. It is frequently membranous and continuous with the dorsal wall of pharynx. It is an organ of taste. E. Mandibles. There is a pair of mandibles. They are the first pair of jaws. They are also called as primary jaws or true jaws. Mandibles articulate with the cranium at 2 points. They are heavily sclerotized. They are toothed on their inner border. There are 2 types of teeth. Distal or sharply pointed and are called incisor or cutting teeth, and proximal teeth are called molar or grinding teeth. They act transversely to bite and grind the food into small fragments. IV. Maxillae. They are paired and more complicated than mandibles. They are called secondary jaws or accessory jaws. At proximal end, the first sclerite cardo joins the maxilla to head. The second sclerite is called stipes, which articulates with cardo. Stipes carries a lateral sclerite called palpifer, which bears a 5-segmented antenna-like maxillary palp. On the distal end of the stipes, there are 2 lobes. The outer lobe is called galea and inner lobe is lacinium, which is toothed. Maxill direct the food into the mouth. They hold the food in place when the mandibles are in action. They act as auxiliary jaws and assist in mastication of food. Sense organs connected with the perception of touch, smell and taste are abundantly found in palpi. V. Hypopharynx. It is a tongue-like organ. It is located centrally in the preoral cavity. Salivary gland duct opens through it. V. Labium, lower lip. It is a composite structure formed by the fusion of 2 primitive segmented appendages. It bounds the mouth cavity from below or behind. It forms the base of the preoral cavity. It consists of 3 median sclerites, viz. Submentum, large basal sclerite, mentum, middle sclerite, and prementum, apical sclerite. On the lateral side of the prementum there are 2 small lateral sclerites called palpigor bearing 3 segmented labial palpi. Distally, prementum bears 2 pairs of lobes. The other pair of lobes is called paraglossae and inner pair of lobes, glossae. Both pairs when fused are called ligula. 2. Piercing and sucking, hemipterous, bug type, e.g. plant bugs. Labium projects downwards from the anterior part of the head like a beak. Beak is 4-segmented and grooved throughout its entire length. At the base of the labium there is a triangular flap-like structure called labrum. Labium is neither involved in piercing nor sucking. It functions as a protective covering for the 4 stylets fascicle found within the groove. Both mandibles and maxillae are modified into long, slender, sclerotized hair-like structure called stylets. They are lying close together and suited for piercing and sucking. The tips of the stylets may have minute teeth for piercing the plant tissue. The inner maxillary stylets are doubly grooved on their inner faces. When these are closely opposed they form 2 canals, viz. Food canal and salivary canal. Through sap and saliva are conducted respectively. Saliva contains enzymes or toxins that can distort plant cell wall to permit the stylets to penetrate down and reach phloem for sucking the sap. Both palps are absent. 3. Piercing and sucking, dipterous, mosquito type, e.g. female mosquito. Mouthparts of female mosquito consists of an elongate labium which is grooved forming a gutter which encloses 6 stylets. The stylets are composed of labrum, epipharynx enclosing the food canal, the hypopharynx containing the salivary canal, 2 maxillae and 2 mandibles. Both the ends of maxillary stylets and mandibular stylets are saw-like and suited piercing flesh. The stylets are inserted into host skin by a strong downward and forward thrust of body. Both mandibles and maxillae are reduced in male and they feed on plant nectar and juices of decaying fruits. Female pierces the skin of human beings into which it injects saliva containing an anticoagulant to keep the blood flowing without clotting, and an anesthetic to keep the victim unaware of the bite and sucks up the blood. Labium does not pierce but folds up or back as stylets pierce. Maxillary palpi are present. 4. Chewing and lapping type, e.g. honeybee. Labrum and mandibles are as in biting and chewing type of mouth parts. But mandibles are blunt and not toothed. They are useful to crush and shape wax for comb building, ingest pollen grains and other manipulative functions. Maxillolabial structures are modified to form the lapping tongue. The tongue unit consists of 2 galea of maxillae, 2 labial palpi and elongated flexible hairy glossa of labium. The glossa terminates into a small circular spoon-shaped lobe called spoon or bowdin or flabellum which is useful to lick the nectar. 5. Rasping and sucking, e.g. thrips. Mouth cone consists of labrum, labium and maxillae. There are 3 stylets derived from 2 maxillae and left mandible. Right mandible is absent. Stylets are useful to lacerate the plant tissue and the oozing sap is sucked up by the mouth cone. Both maxillary palpi and labial palpi are present. 6. Mandibulosectorial type, e.g. grub of antlion. Mandibles are elongate sickle-shaped and grooved on the inner surface. Each maxilla is elongated and fits against the mandibular groove to form a closed food canal. The body of the insect victim is pierced by the opposing mandibles and fluids are extracted. 7. Sponging type, e.g. housefly. The proboscis is fleshy, elbowed, retractile and projects downwards from head. The proboscis can be differentiated into basal rostrum and distal haustellum. The proboscis consists of labium which is grooved on its anterior surface. Within this groove lie the labrum epipharynx, enclosing the food canal, and slender hypopharynx containing the salivary canal. Mandibles are absent. Maxillae are represented by single-segmented maxillary palpi. The end of the proboscis is enlarged, sponge-like and two-lobed, which acts as suction pads. They are called oral discs or labella. The surfaces of labella are transferred by capillary canals called pseudotracheae, which collect the liquid food and convey it to the canal. Labella function as sponging organs and are capable of taking exposed fluids. These insects often spit enzyme-containing saliva onto solid foods to liquefy them. 8. Siphoning type, e.g. moths and butterflies. Mouth parts consists of elongate sucking tube or proboscis. It is formed by two greatly elongated galleae of maxillae which are zippered together by interlocking spines and hooks. Galleae are grooved on their inner surface, and when they are fitting together closely they form a suctorial food canal through which the nectar is sucked up. The proboscis is coiled up like watch spring and kept beneath the head when it is not in use. By pumping of blood into galleae, the proboscis is extended. The other mouth parts are reduced or absent except the labial palpi and smaller maxillary palpi. Lecture 5. Insect thorax. Segmentation of thorax. Leg structure and its modifications. Thorax. The insect thorax is divided into three parts. The prothorax pro equals first, mesothorax, meso equals middle, and metathorax meta equals last. Each segment consists of hardened plates, or sclerites. Dorsal sclerites are called noda sing. Nodum, lateral sclerites are called pleura sing. Pleuron and ventral sclerites are called sterna sing. Sternum. Each of the three thoracic segments contains one pair of legs. Wings are found only on the meso and metathoracic segments. Legs. The fore legs are located on the prothorax, the mid legs on the mesothorax, and the hind legs on the metathorax. The fore legs are located on the prothorax, the mid legs on the mesothorax, and the hind legs on the metathorax. Each leg has 6 major components, listed here from proximal to distal: coxa P1, coxae trochanter, femur P1, femora tibia P1, tibi tarsus P1, tarsi protarsus. The femur and tibia may be modified with spines. The tarsus appears to be divided into 1 to 5 pseudosegments, called tarsomers. Structure. In almost all insects, all the three thoracic segments viz. Pro, meso and metathorax bear a pair of segmented legs. Each leg consists of 5 segments viz. Coxa, trochanter, femur, tibia and tarsus. Coxa, place. Coxae, it is the first or proximal leg segment. It articulates with the cup-like depression on the thoracic pleuron. It is generally freely movable. Trochanter, it is the second leg segment. It is usually small and single-segmented. Trochanter seems to be two-segmented in dragonfly, dameselfie and ichneummonid wasp. The apparent second trochanter is in fact a part of femur, which is called trochantellus. Femur, place. Femora, it is the largest and stoutest part of the leg and is closely attached to the trochanter. Tibia, place. Tibi, it is usually long and provided with downward projecting spines which aid in climbing and footing. Tibia of many insects is armed with large movable spur near the apex. Tarsus, place. Tarsi, it is further subdivided. The sub-segment of the tarsus is called tarsomere. The number of tarsomers vary from 1 to 5. The basal tarsal segment is often larger than others and is named as basitarsus. Protarsus. Beyond the tarsus there are several structures collectively known as protarsus. Tarsus terminates in a pair of strongly curved claws with 1 or 2 pads of cushions at their base between them. A median pad between the claws is usually known as aurolium, and a pair of pads at their base are called pulvili, pulvilis singular. Leg pads are useful while walking on smooth surface, and claws give needed grip while walking on rough surface. Leg pads are useful while walking on smooth surface, and claws give needed grip while walking on rough surface. When one structure is used, the other is bent upwards. When one structure is used, the other is bent upwards. Leg types and function. Cursorial, used for walking, running. Some textbooks distinguish the two by calling walking legs ambulatory or gressorial, but the leg structure is basically the same. Raptorial, 4 legs modified for grasping. These are often associated with praying mantids. Fossorial, 4 legs and tibi specialized for digging, common in ground-dwelling insects. Saltatorial, hind legs adapted for jumping, characterized by an elongated femur and tibia. Pronotum is the dorsal sclerite of the prothorax, which can be highly modified in various groups such as the Homoptera, Blattaria, and Coleoptera. Types of legs. Insects are 6-legged arthropods, and hence the class is also called hexapoda. In insects, legs perform varied functions and are modified accordingly. 1. Digging or fossorial type. The forelegs are greatly expanded. Tibia is digitate with 3-segmented tarsus beneath. The legs are used for digging soil, e.g., mole cricket. 2. Jumping or saltatorial type. The hindlegs are modified for leaping or jumping. Femur is greatly enlarged. Tibia is very long, e.g., grasshopper. Natatorial. 4 or hind legs adapted for swimming, characterized by elongated setae on tarsi 3. Walking or running type. All 3 pairs of legs are equal in size and comparatively long. Trochanter is 2-segmented, e.g., cockroach. 4. Grasping or raptorial type. The forelegs are modified for catching prey. The coxae are elongate and movable. The femora are spiny and grooved along the lower side. The tibia are also spiny and fit into the groove along the femur. The prey is held between the femur and tibia. Tarsus is 5-segmented, e.g., praying mantids. 5. Swimming or natatorial type. Usually the hind legs are modified for swimming. Hind coccyx are flat and fixed to the body. Numerous long stiff hairs are present on the lateral aspects of the tibia and tarsus, e.g. diving beetles. 6. Pollen carrying type. The hind legs of honeybees are modified for carrying pollen. At the junction of tibia and basitarsus, a cavity guarded by hairs is present which is used for carrying pollen. This structure is also called carbiculum, e.g., worker honeybee. 7. Antenna cleaner. The forelegs of honeybees are modified for cleaning antenna. The first segment of tarsus has a notch, which can be closed by the flat tibial spur. The notch has an inner lining of fine hairs. The antenna is placed in the notch, closed by the spur and then drawn out to clean, e.g., worker honeybees. 8. Clinging type. The legs are strong and adapted for maintaining a strong and firm hold on the host. Tarsi are single-segmented and terminate in a single sickle-shaped claw which works against a tibial process, e.g., head louse and body louse. 9. Climbing type. The terminal segment of the leg, protarsus, bears 2 claws and beneath the claws are 2 lobes called pulvuli. Between the pulvuli is an elongate spine called empodium. The empodium and pulvuli help the insect to climb smooth surfaces, e.g. housefly. Lecture 6. Insect wing, structure and its modifications. Wings. Insects have evolved many variations of the wing. Wing venation is a commonly used taxonomic character, especially at the family and species level. Membranous wings are thin and more or less transparent. This type of wings is found among the Odonata and Neuroptera. Halteres are an extreme modification among the order Diptera, true flies, in which the hind wings are reduced to mere nubs used for balance and direction during flight. Haltere. Elytra thin. Elytron are the hardened, heavily sclerotized forewings of beetles. Elytron are the hardened, heavily sclerotized forewings of beetles, order Coleoptera, and are modified to protect the hind wings when at rest. A variation of the elytra is the hemelytra. The forewings of hemipterans are said to be hemelytrous because they are hardened throughout the proximal 2/3, approximately, while the distal portion is membranous. Unlike elytra, hemelytra function primarily as flight wings. In both cases, the membranous hind wings, when present, are used in flight and are folded beneath the forewings when at rest. The wings of butterflies and moths are covered with scales, and mosquitoes possess scales along wing veins. Neopterous versus Paleopterous wing conditions. In most living insects, the Neoptera, there are 3 axillary sclerites that articulate with various parts of the wing. In the Neoptera, a muscle on the 3rd axillary causes it to pivot about the posterior nodal wing process and thereby to fold the wing over the back of the insect. In some groups of Neoptera, such as butterflies, the ability to fold the wings over the back has been lost. 2 orders of winged insects, the Ephemeroptera and Odonata, have not evolved this wing flexing mechanism, and their axillary sclerites are arranged in a pattern different from that of the Neoptera. These 2 orders, together with a number of extinct orders, form the Paleoptera. Types of insect wings. Among invertebrate animals, only insects possess wings. Wings are present only in adult stage. Number of wings vary from 2 pairs to none. Certain primitive insects like silverfish and springtail have no wings, Apteris. Ectoparasites like head louse, poultry louse and flea are secondarily wingless. Wings are deciduous in ants and termites. There is only 1 pair of wings in the true flies. Normally, 2 pairs of wings are present in insects and they are borne on pterothoracic segments viz. Mesothorax and metathorax. Wings are moved by thoracic flight muscles attached to their bases. Wing is a flattened double-layered expansion of body wall with a dorsal and ventral lamina having the same structure as the integument. Both dorsal and ventral Paleopterous. Laminae grow, meet and fuse except along certain lines. Thus a series of channels is formed. These channels serve for the passage of tracheae, nerves and blood. Wing is nourished by blood circulating through veins. Later the walls of these channels become thickened to form veins or nervures. The arrangement of veins on the wings is called venation, which is extensively used in insect classification. The principal longitudinal veins arranged in order from the anterior margin are costa C, subcosta SC, radius R, median M, cubitus Q, and anal veins A. Small veins often found interconnecting the longitudinal veins are called cross veins. Due to the presence of longitudinal veins and cross veins, the wing surface gets divided into a number of enclosed spaces termed cells. In insects like dragonfly and dancefly, there is an opaque spot near the costal margin of the wing called pterostigma. Margins and angles. The wing is triangular in shape and has therefore 3 sides and 3 angles. The anterior margin strengthened by the costa is called costal margin and the lateral margin is called apical margin and the posterior margin is called anal margin. The angle by which the wing is attached to the thorax is called humeral angle. The angle between the costal and apical margins is called apical angle. The angle between apical and anal margins is anal angle. Wing regions. The anterior area of the wing supported by veins is usually called remigium. The flexible posterior area is termed vanus. The 2 regions are separated by vanal fold. The proximal part of vanus is called jugum, when well developed is separated by a jugal fold. The area containing wing articulation sclerites, pterolia is called axilla. Wing types. 1. Tegmina, singular, tegmen, wings are leathery or parchment-like. They are protective in function. They are not used for flight. E.g., forewings of cockroach and grasshopper. 2. Elytra. Singular, elytron, the wing is heavily sclerotized. Wing venation is lost. Wing is tough and it is protective in function. It protects hind wings and abdomen. It is not used during flight. But during flight they are kept at an angle allowing free movement of hind wings. E.g., forewings of beetles and weevils. 3. Hemelytra. Singular, hemeletron, the basal half of the wing is thick and leathery and distal half is membranous. They are not involved in flight and are protective in function. E.g., forewing of heteropteran bugs. 4. Halteres. Singular, halter, in true flies the hind wings are modified into small knobbed vibrating organs called halter. Each halter is a slender rod clubbed at the free end capitellum, and enlarged at the base scabellum. On the basal part 2 large group of sensory bodies forming the smaller Hicks papillae and the large set of scapal plate. They act as balancing organs and provide the needed stability during flight. E.g. true flies, mosquito, male scale insect. 5. Fringed wings. Wings are usually reduced in size. Wing margins are fringed with long setae. These insects literally swim through the air. E.g., thrips. 6. Scaly wings. Wings of butterfly and moths are covered with small colored scales. Scales are unicellular flattened outgrowth of body wall. Scales are inclined to the wing surface and overlap each other to form a complete covering. Scales are responsible for color. They are important in smoothing the airflow over wings and body. 7. Membranous wings. They are thin, transparent wings and supported by a system of tubular veins. In many insects either forewings, true flies, or hindwings, grasshopper, cockroach, beetles and earwig, or both forewings and hindwings, wasp, bees, dragonfly and damselfly, are membranous. They are useful in flight. Wing coupling. Among the insects with 2 pairs of wings, the wings may work separately as in the dragonflies and damselflies. But in higher pterygote insects, fore and hind wings are coupled together as a unit, so that both pairs move synchronously. By coupling the wings the insects become functionally 2-winged. Types of wing coupling. 1. Hamulate. A row of small hooks is present on the costal margin of the hind wing which is known as hamuli. These engage the folded posterior edge of forewing. E.g. bees. 2. Amplexiform. It is the simplest form of wing coupling. A linking structure is absent. Coupling is achieved by broad overlapping of adjacent margins. E.g. butterflies. 3. Frenate. There are 2 subtypes. E.g. fruit-sucking moth. I. Male frenate. Hindwing bears near the base of the costal margin a stout bristle called frenulum which is normally held by a curved process, retinaculum arising from the subcostal vein found on the surface of the forewing. E. Female frenate. Hindwing bears near the base of the costal margin a group of stout bristle frenulum, which lies beneath extended forewing and engages there in a retinaculum formed by a patch of hairs near cubitus. 4. Jugate. Jugum of the forewings are lobe-like and it is locked to the costal margin of the hindwings. E.g. Hepialid moths. Lecture 7. Insect abdomen. Structure and its modifications. Abdomen. The dorsal and ventral abdominal segments are termed terga, singular tergum, and sterna, singular sternum, respectively. Spiracles usually can be found in the conjunctive tissue between the terga and sterna of abdominal segments 1 to 8. Reproductive structures are located on the 9th segment in males, including the edagus, or penis, and often a pair of claspers, and on the 8th and 9th abdominal segments in females, female external genitalia, copulatory openings and ovipositor. Modifications of the ovipositor. Sexual dimorphism. Female, note the long ovipositor, male, 2 cerci at the end of between the cerci, the abdomen. Abdominal structures in insects. Basic structures. Segmentation is more evident in abdomen. The basic number of abdominal segments in insect is 11 plus a telson which bears anus. Abdominal segments are called uromers. On 8th and 9th segment of female and 9th segment of male, the appendages are modified as external organs of reproduction or genitalia. These segments are known as genital segments. Usually 8 pairs of small lateral openings, spiracles, are present on the first 8 abdominal segments. In grasshoppers, a pair of tympanum is cerci. Ovipositor found 1 on either side of the first abdominal segment. It is an auditory organ. It is obliquely placed and connected to the metathoracic ganglia through auditory nerve. Modifications. Reduction in number of abdominal segments has taken place in many insects. In springtail only 6 segments are present. In housefly only segments 2 to 5 are visible and segments 6 to 9 are telescoped within others. In ants, bees and wasps, the first abdominal segment is fused with the metathorax and is called propydium. Often the second segment forms a narrow petiole. The rest of the abdomen is called gaster. In queen termite after mating the abdomen becomes gradually swollen due to the enlargement of ovaries. The abdomen becomes bloated and as a result sclerites are eventually isolated as small islands. Obesity of abdomen of queen termite is called physogastry. Abdominal appendages. I. Progenital abdominal appendages in wingless insects. 1. Styli. Stylus, singular, varying number of paired tube-like outgrowths are found on the ventral side of the abdomen of silverfish. These are reduced abdominal legs which help in locomotion. 2. Coliphore or ventral tube or glue peg. It is located on the ventral side of the first abdominal segment of springtail. It is cylindrical. It is protruded out by the hydrostatic pressure of hemolymph. It might serve as an organ of adhesion. It aids in water absorption from the substratum and also in respiration. 3. Retinaculum or tenaculum or catch. It is present on the ventral side of the third abdominal segment. It is useful to hold the springing organ when not in use. 4. Furcula or furca. This is a Y-shaped organ. It is present on the venter of fourth abdominal segment. When it is released from the catch, it exerts a force against the substratum and the insect is propelled in the air. Abdominal appendages in immature insects. 1. Tracheal gills. Gills are lateral outgrowths of body wall which are richly supplied with tracheae to obtain oxygen from water in naiads, aquatic immature stages of hemimetabolous insects. 7 pairs of filamentous gills are present in the first 7 abdominal segments of naiads of mayfly and are called as lateral gills. 3 or 2 leaf-like gills, lamellate, are found at the end of abdomen of naiad of damselfly and are called as caudal gills. In dragonfly the gills are retained within the abdomen in a pouch-like rectum and are called as rectal gills. 2. Anal papillae. A group of 4 papillae surrounds the anus in mosquito larvae. These papillae are concerned with salt regulation. 3. Dolichasters. These structures are found on the abdomen of antlion grub. Each dolichaster is a segmental protuberance fringed with setae. 4. Prolobes. These are present in the larva of moth, butterfly and sawfly. 2 to 5 pairs are normally present. They are unsegmented, thick and fleshy. The tip of the proleg is called planta upon which are borne heavily sclerotized hooks called crochets. They aid in crawling and clinging to surface. Abdominal appendages in winged adults. 1. Cornicles. Aphids have a pair of short tubes known as cornicles or siphonculi projecting from dorsum of 5th or 6th abdominal segment. They permit the escape of waxy fluid which perhaps serves for protection against predators. 2. Caudal breathing tube. It consists of 2 grooved filaments closely applied to each other forming a hollow tube at the apex of abdomen. E.g. water scorpion. 3. Cerci. Circus singular. They are the most conspicuous appendages associated normally with the 11th abdominal segment. They are sensory in function. They exhibit wide diversity in form. Long and many segmented, e.g. mayfly. Long and unsegmented, e.g. cricket. Short and many segmented, e.g. cockroach. Short and unsegmented, e.g. grasshopper. Sclerotized and forceps-like, e.g. earwig. Cerci are useful in defense, prey capture, unfolding wings and courtship. Asymmetrical cerci, male embiid. Left circus is longer than right and functions as clasping organ during copulation. 4. Median caudal filament. In mayfly and also in a wingless insect silverfish, the epiprocta is elongated into circus-like median caudal filament. 5. Pygostyles. A pair of unsegmented cerci-like structures are found in the last abdominal segment of scalid wasp. 6. Anal styli. A pair of short unsegmented structure found at the end of the abdomen of male cockroach. They are used to hold the female during copulation. 7. Ovipositor. The egg-laying organ found in female insect is called ovipositor. It is suited to lay eggs in precise microhabitats. It exhibits wide diversity in form. Short and horny, e.g. short-horned grasshopper. Long and sword-like, e.g. katydid, long-horned grasshopper needle-like, e.g. cricket. Ovipositor modified into sting, e.g. worker honeybee. Pseudovipositor. An appendicular ovipositor is lacking in fruit flies and house flies. In fruit flies, the elongated abdomen terminates into a sharp point with which the fly pierces the rind of the fruit before depositing the eggs. In the housefly the terminal abdominal segments are telescopic and these telescopic segments aid in oviposition. The ovipositor of housefly is called pseudovipositor or ovitubus or oviscapt. Male genitalia. External sexual organs of male insects are confined to 9th abdominal segment. In damselfly, the functional copulatory organ is present on the venter of 2nd abdominal segment. Lecture 8. Anatomy. Digestive system. Digestive system. The digestive system, sometimes referred to as the alimentary canal, should be easily seen in the dissected specimens. It is a long tube-like structure that runs from the mouth to the anus and is centrally located within the body cavity, or hemocoel. The anteriormost region is called the foregut, or stomodium, which includes the buccal cavity, the esophagus, and the crop. The primary function of the foregut is to begin the breakdown of food particles and transport them to the next region, the midgut or mesenteron. The midgut is the major area of digestion and absorption. Undigested food particles then pass into the third region, the hindgut or proctidium, which consists of the ileum, colon, rectum, and often rectal pads. The hindgut functions in water and solute reabsorption and waste excretion. The 3 sections of the digestive tract can be easily identified by structures found at the junction of each region. Gastric cc, for example, mark the end of the foregut and beginning of the midgut. It is believed that the purpose of these structures is to increase surface area for greater nutrient absorption. The constriction at the gastric cc also marks the spot of the cardiac valve or sphincter. The alimentary canal of insects is a long, muscular and tubular structure extending from mouth to anus. It is differentiated into 3 regions viz. Foregut, midgut and hindgut. 1. Foregut. It is ectodermal in origin. Anterior invagination of ectoderm forms foregut stomodium. Internal cuticular lining is present. Terminal mouthparts leads into a preoral cavity. Preoral cavity between epipharynx and hypopharynx is called a sebareum. Preoral cavity between hypopharynx and salivary duct is salivarium. Behind the mouth a well-musculated organ called pharynx is present which pushes the food into oesophagus. Pharynx acts as a sucking pump in sap feeders. Oesophagus is a narrow tube which conduct food into crop. Crop is the dilated distal part of esophagus acting as food reservoir. In bees crop is called as honey stomach where nectar conversion occurs. Proventriculus or gizzard is the posterior part of foregut and is musculated. It is found in solid feeders and absent in fluid feeders or sap feeders. Food flow from foregut to midgut is regulated through cardial or esophageal valve. The internal cuticle of gizzard is variously modified as follows. I. Teeth like in cockroach to grind and strain food. E. Plate-like in honeybee to separate pollen grains from nectar. E. Spine-like in flea to break the blood corpuscles. 2. Midgut. It is endodermal in origin and also called as mesentery. This part contains no cuticular lining. Midgut is made up of 3 types of epithelial cells. I. Secretory cells, columnar cells. E. Goblet cells, aged secretory cells. E. Regenerative cells which replaces secretory cells. Important structures present in midgut are as follows. A. Paratrophic membrane. It is the internal lining of midgut, secreted by anterior or entire layer of midgut epithelial cells. Present in solid feeders and absent in sap feeders. This layer is semi-permeable in nature to digestive juices and digestion products. It lubricate and facilitate food movement. Envelops the food and protects the midgut epithelial cells against harder food particles. E. Gastric cc. Enteric cc or hepatic cc, finger-like outgrowths found in anterior or posterior ends of midgut. This structure increases the functional area of midgut and shelter symbiotic bacteria in some insects. E. Pyloric valve. Proctodel valve. Midgut opens into hindgut through pyloric valve, which regulate food flow. In certain immature stages of insects, midgut is not connected to hindgut till pupation. E.g., honeybee grub. IV filter chamber. It is a complex organ in which 2 ends of ventriculus and the beginning of hindgut are enclosed in a sac. This is useful to short-circuit excess water found in liquid food in homopteran insects. This process avoids dilution of digestive enzymes and concentrates food for efficient digestion. Also helps in osmoregulation by preventing dilution of hemolymph. 3. Hindgut. It is ectodermal in origin and produced by the posterior invagination of ectoderm. Internal cuticular lining is present, which is permeable to salts, ions, amino acids and water. The main functions of hindgut are the absorption of water, salt and other useful substances from the feces and urine. Hindgut is differentiated into 3 regions viz. Ileum, colon and rectum. In the larvae of scarabids and termites, ileum is pouch-like for housing symbionts and acts as fermentation chamber. Rectum contains rectal pads helping in dehydration of feces and it opens out through anus. Gut physiology. Primary functions of the gut is to digest the ingested food and to absorb the metabolites. Digestion process is enhanced with the help of enzymes produced by digestive glands and microbes housed in special cells. Digestive glands. A. Salivary glands. In cockroach, a pair of labial glands acts as salivary gland where the salivary ducts open into salivarium. In caterpillars, mandibular glands are modified to secrete saliva, where the salivary glands are modified for silk production. Functions of saliva. 1. To moisten and to dissolve food. 2. To lubricate mouthparts. 3. To add flavor to gustatory receptors. 4. In cockroach, the saliva contains amylase for the digestion of starch. 5. In honeybee, saliva contains invertase for sucrose digestion. 6. In jacid, saliva contains lipase and protease for lipids and protein digestion. Jacid saliva also contains toxins which produces tissue necrosis and phytotoxemia on the plant parts. 7. In plant bug, saliva contains pectinase which helps in stylet penetration and extra-intestinal digestion. 8. In mosquito, saliva contains anticoagulant which prevents blood clotting. 9. In gall-producing midges, saliva contains indole acetic acid, IAA. 10. In disease-transmitting ectors, the saliva paves way for the entry of pathogens. B. Hepatic cc and midgut epithelial cells. It secretes most of the digestive juices. 2 types of cells were involved in the enzyme secretion. Holocrine. Epithelial cells disintegrate in the process of enzyme secretion. Merocrine. Enzyme secretion occurs without cell breakdown. Digestive enzymes. Insect group enzyme substrate. Phytophagus larva amylase starch maltase maltose invertase sucrose omnivorous insects protease protein lipase lipid nectar feeders invertase sucrose wood boring insects and termites cellulase cellulose. Meat-eating maggots collagenase collagen and elastin bird lice keratinase keratin. C. Microbes in digestion. In the insect body, few cells were housing symbiotic microorganisms called as mycetocyte. These mycetocytes aggregate to form an organ called mycetome. I. Flagellate protozoa. It produces cellulase for cellulose digestion in termites and wood cockroach. E. Bacteria. It helps in wax digestion in wax moth. E. Bedbug and cockroach obtain vitamin and amino acids from microbes. These microbes were transmitted between individuals through food exchange, mouth-to-mouth feeding, called trophallaxis, and through egg called as transovarial transmission. In plant bug and antlion grub, partial digestion occurs in the host body prior to food ingestion called as extra-intestinal digestion. In most of the insects, digestion occurs in midgut. Absorption. In many insects, absorption of nutrients occurs through microvilli of midgut epithelial cells by diffusion. Absorption of water and ions occur through rectum. In cockroach, lipid absorption occurs through crop. In termites and scarabids, white grubs, absorption occurs through ileum. In solid feeders, resorption of water from the feces occurs in the rectum and the feces is expelled as pellets. In sap feeders, liquid feeders, the feces is liquid-like. The liquid feces of homopteran bugs, aphids, mealy bugs, scales and psyllids with soluble sugars and amino acids is known as honeydew, which attracts ants for feeding. Lecture 9. Anatomy. Excretory system. Excretory system. Removal of waste products of metabolism, especially nitrogenous compounds from the body of insects, is known as excretion. The excretion process helps the insect to maintain salt-water balance and thereby physiological homeostasis. Following are the excretory organs. Near the junction of the midgut and hindgut are long, thin structures called Malpighian tubules. These range in number from a few to hundreds, but only aphids, order Homoptera, are currently known to have none. Malpighian tubules are creamy to yellow in color and work in conjunction with the ileum to provide the primary site for osmoregulation and excretion. 1. Malpighian tubules. Thin, blind-ending tubules, originating near the junction of mid and hindgut, predominantly involved in regulation of salt, water and nitrogenous waste excretion. This structure was discovered by Marcello Malpighi. 2. Nephrocytes, cells that sieve the hemome for products that they metabolize, pericardial cells. 3. Fat bodies, a loose or compact aggregation of cells, mostly tryphocytes, suspended in the hemocoel, responsible for storage and excretion. 4. Enocytes, these are specialized cells of hemocoel, epidermis or fat body with many functions. One of the function is excretion. 5. Integument, the outer covering of the living tissues of an insect. 6. Tracheal system, the insect gas exchange system, comprising tracheae and tracheoles. 7. Rectum, the posterior part of hindgut. Among the above organs, Malpighian tubules are the major organ of excretion. Excretion and osmoregulation. Insect feces, either in liquid form or solid pellets, contains both undigested food and metabolic excretions. Aquatic insects excrete dilute wastes from their anus directly into water by flushing with water. But, terrestrial insects must conserve water. This requires efficient waste disposal in a concentrated or even dry form, simultaneously avoiding the toxic effects of nitrogen. Both terrestrial and aquatic insects must conserve ions, such as sodium Na+, potassium K+, and chloride Cl-, that may be limiting in their food or lost into the water by diffusion. Therefore, the production of insect excreta, urine or pellets, is a result of two related processes: excretion and osmoregulation, maintenance of favorable osmotic pressure and ionic concentration of body fluid. The system responsible for excretion and osmoregulation is referred to as excretory system, and its activities are performed largely by the Malpighian tubules and hindgut. However, in freshwater insects, hemolymph composition is regulated in response to loss of ions to the surrounding water, with the help of excretory system and special cells. Special cells are called chloride cells, which are present in the hindgut, capable of absorbing inorganic ions from the dilute solutions, e.g., naids of dragonflies and damselflies. Malpighian tubules. The main organ of excretion and osmoregulation in insects are the Malpighian tubules, acting in association with rectum or ileum. Malpighian tubules are outgrowths of the alimentary canal and consist of long thin tubules formed of a single layer of cells surrounding a blind-ending lumen. They are absent in springtail and aphids, 2 numbers in scale insects, 4 in bugs, 5 in mosquitoes, 6 in moths and butterflies, 60 in cockroach and more than 200 in locusts. Generally they are free, waving around in the hemolymph where they filter out solutes. Each tubule is externally covered by peritoneal coat and supplied with muscle fibers, aiding in peristalsis and trachealose. Functional differentiation of the tubules was seen, with the distal secretory region and proximal absorptive region. Physiology. The Malpighian tubules produce a filtrate, the primary urine, which is isosmotic but ionically dissimilar to the hemolymph and selectively reabsorbs water and certain solutes, but eliminates others. The Malpighian tubules produces an iosmotic filtrate which is high in K+ and low in Na+, with Cl- as major anion. The active transport of ions, especially K+ into the tubule lumen, generates an osmotic pressure gradient for the passive flow of water. Sugars and most amino acids are also passively filtered from the hemolymph via junctions between the tubule cells, whereas amino acids and non-metabolizables and toxic organic compounds are actively transported into the tubule lumen. Sugar is resorbed from the lumen and returned to the hemolymph. The continuous secretory activity of each Malpighian tubule leads to a flow of primary urine from its lumen towards and into the gut. In the rectum, the urine is modified by removal of solutes and water to maintain fluid and ionic homeostasis of the body. Nitrogenous excretion. Terrestrial insects excrete waste products as uric acid or certain of its salts called urates, which were water insoluble and requires less amount of water for waste product removal. This type of excretion is known as urealism. In aquatic insects, ammonia is the excretory product, which is freely soluble in water and requires more amount of water for waste product removal. This type of excretion is known as ammonotelism. Cryptonephry. The distal ends of the Malpighian tubules are held in contact with the rectal wall by the paranephric membrane, which is concerned either with efficient dehydration of feces before their elimination or ionic regulation, e.g., adult Coleptera, larval Lepidoptera and larval Symphyta. Functions of Malpighian tubule. Excretory in function, mainly concerned with removal of nitrogenous wastes. The other accessory functions are as follows. 1. Spittle secretion in spittle bug. 2. Light production in Boletophila. 3. Silk production in larval Neuroptera. Storage excretion. The excretory waste materials are retained within the body in different sites. I. Uric acid is stored as urates in the cells of fat body, e.g., American cockroach. E. Uric acid is stored in the body wall, giving white color, e.g., red cotton bug. E. Uric acid is stored in the male accessory glands to produce the outer coat of spermatophore, which is excreted during copulation. IV. Uric acid is stored in the wing scales, giving white color, e.g., Pierid butterflies. V. Waste products of pupal metabolism, meconium, is stored and released during adult emergence. Lecture 10. Anatomy. Respiratory system. Respiratory system. The insect respiratory system is made up of a series of tubes that originate from spiracles, openings of the exoskeleton that allow for gas exchange, and extend throughout the body. Internally, the tubes, or trachea, appear as thin white lines throughout the hemocoel and are particularly noticeable surrounding internal organs. Trachea deliver oxygen to internal organs and tissues. Compare the tracheae with the Malpighian tubules. They are often very similar in appearance. Did you confuse tracheae with Malpighian tubules earlier? 2 ways to distinguish the structures are color and location. Tracheae have a shinier appearance under the scope and may even appear silvery. As for location, Malpighian tubules are found at the junction of the midgut and hindgut, although they may extend outward into the hemocoel, whereas tracheae are positioned throughout the body. Respiratory system. Similar to aerobic animals, insects must obtain oxygen from their environment and eliminate carbon dioxide respired by their cells. This is gas exchange through series of gas-filled tubes providing surface area for gaseous exchange. Respiration strictly refers to oxygen-consuming, cellular metabolic processes. Air is supplied directly to the tissue and hemolymph. Blood is not involved in the respiratory role. Gas exchange occurs by means of internal air-filled tracheae. These tubes branch and ramify through the body. The finest branch is called tracheole, contact all internal organs and tissues and are numerous in tissues with high oxygen requirements. Air usually enters the tracheae via specular openings positioned laterally on the body. No insect has more than 10 pairs, 2 thoracic and 8 abdominal. Based on the number and location of functional spiracles, respiratory system is classified as follows. 1. Polypneistic, 10 pairs, 2 in thorax and 8 in abdomen. E.g. grasshopper. 2. Hemipneistic, out of 10 pairs, 1 or 2 non-functional. 3. Peripneistic, 9 pairs, 1 in thorax, 8 in abdomen, e.g. caterpillar. 4. Amphipneistic, 2 pairs, 1 anterior, 1 posterior, e.g. maggot. 5. Propneistic, 1 pair anterior pair, e.g. puparium. 6. Metaneustic, 1 pair posterior pair, e.g. wriggler. 7. Hypopneistic, 10 pairs, 7 functional, 1 thorax plus 6 abdominal, 3 non-functional, e.g. head louse. 8. Apneustic, all spiracles closed tracheal system, e.g. naiad of mayfly. Organs of respiration. Spiracles. Spiracles have a chamber or atrium with an opening and closing mechanism called atrial valve. This regulate air passage and minimize water loss. Each spiracle is set in a sclerotized cuticular plate called a peritreme. Tracheae are invaginations of the epidermis and thus their lining is continuous with the body cuticle. The ringed appearance of the tracheae is due to the spiral ridges called taenidia. This allow the tracheae to be flexible but resist compression. The cuticular linings of the tracheae are shed during molting. Tracheoles are less than 1 micrometer in diameter and they end blindly and closely contact the respiring tissues. Taenidia and wax layer is absent. Cuticulin layer is permeable to gases. It is intracellular in nature, but enclosed only in the cytoplasm of tracheal cell called tracheoblast. Gaseous exchange occurs across tracheoles. There are 4 tracheal trunks, viz. Lateral, dorsal, ventral and visceral, helping in the passage of air. In the trachea, thin-walled collapsible sac-like dilations are present, called as air sacs where taenidia is absent. Air sacs acts as oxygen reservoir. Provide buoyancy to flying and aquatic insects. Provide space for growing organs. Acts as sound resonator and heat insulators. Mechanism of respiration. Oxygen enters the spiracle and passes through the length of the tracheae to the tracheoles and into the target cells by a combination of ventilation and diffusion along a concentration gradient, from high in the external air to low in the tissue. Whereas the net movement of oxygen molecules in the tracheal system is inward, inspiration, the net movement of CO2 and water vapor molecules is outward, expiration. Respiration in aquatic insects. 1. Closed tracheal system. In some aquatic and many endoparasitic larvae, spiracles are absent and the tracheae divide peripherally to form a network. This covers the body surface, allowing cutaneous gas exchange. E.g. gills, tracheated thin outgrowth of body wall. Lamellate gills, mayfly naiad. Filamentous gills, damselfly naiad. Rectal gills, dragonfly naiad. 2. Open tracheal system. I. Air store. Air bubble stored beneath wings acts as physical gill, e.g. water bug. E. Respiratory siphon, e.g. wriggler. E. Caudal breathing tube, e.g. water scorpion. IV. Plastron. Closely set hydrofuge hairs of a picuticle hold a thin film of air indefinitely. Lecture 11. Anatomy. Circulatory system. Circulatory system. Unlike the closed circulatory system of humans, insect circulatory systems are said to be open, meaning that they lack a complex network of veins and arteries to help transport blood throughout the body. Instead, insect blood called hemolymph flows relatively freely throughout the hemocoel. Figure 8-2. Circulatory system. Arrows indicate direction of flow of hemolymph. Only one vessel is present in the insect circulatory system, the dorsal vessel. Posteriorly, in the abdominal region, the dorsal vessel acts as the heart, pumping hemolymph forward into the anterior region, in the head and thorax where it acts as the aorta and dumps the hemolymph into the head. It flows posteriorly and is returned to the heart via ostia, which are small slits in the heart region of the dorsal vessel designed for hemolymph uptake. To view the dorsal vessel, examine the back, or dorsal region, of the insect's body cavity for a very thin line that runs longitudinally from the head to the tip of the abdomen. Use the grasshopper or specimen that was ventrally dissected, as dorsal dissections will likely mutilate the vessel. Do not be discouraged if you have trouble finding it on your specimen. The dorsal vessel is very, very thin. Compare your specimen to those of your classmates. Circulatory system in insects. Aorta portion of dorsal vessel. Heart portion of dorsal vessel. Circulation in insects is maintained by a system of muscular pumps moving hemolymph through compartments separated by fibromuscular septal membranes. The main pump is the pulsatile dorsal vessel. The anterior part may be called aorta and the posterior part the heart. The dorsal vessel is a simple tube, generally composed of one layer of myocardial cells and with segmentally arranged openings called ostia. The ostia permit the one-way flow of hemolymph into the dorsal vessel due to valves that prevent backflow. There may be up to 3 pairs of thoracic ostia and 9 pairs of abdominal ostia. The dorsal vessel lies in the pericardial sinus, a compartment above a dorsal diaphragm, a fibromuscular septum, a separating membrane, formed of connective tissue and segmental pairs of alary muscles. The alary muscles support the dorsal vessel, but their contractions do not affect heartbeat. Hemolymph enters the periocardial sinus via segmental openings in the diaphragm and then moves into the dorsal vessel via the ostia during a muscular relaxation phase. Waves of contraction start at the posterior end of the body, pump the hemolymph forward in the dorsal vessel and out via the aorta into the head. Next, the appendages of the head and thorax are supplied with hemolymph as it circulates postventrally and finally returns to the pericardial sinus and dorsal vessel. Another important component of the insect circulatory system is the ventral diaphragm, a fibromuscular septum that lies in the floor of the body cavity associated with the ventral nerve cord. Circulation of the hemolymph is aided by active peristaltic contractions of the ventral diaphragm, which direct the hemolymph backwards and laterally in the perineural sinus below the diaphragm. These movements are important in insects that use the circulation in thermoregulation. Ventral diaphragm also facilitates rapid exchange of chemicals between the ventral nerve cord and the hemolymph. Hemolymph is generally circulated to appendages unidirectionally by various tubes, septa, valves and pumps. The muscular pumps are termed accessory pulsatile organs and occur at the base of the antennae and legs. Antennal pulsatile organs releases neurohormones that are carried to the antennal lumen to influence the sensory neurons. Circulation occurs in the wings of young adult. In wing, circulation is sustained by influxes of air into the wing veins, rather than any pulsatile organs. Pulses of air in the fine tracheal tubes of the veins push the hemolymph through the enclosed space of the veins. The insect circulatory system shows high degree of coordination between dorsal vessel, fibromuscular diaphragms and accessory pumps. Hemolymph and its functions. Hemolymph is a watery fluid containing ions, molecules and cells. It is often clear and colorless but may be variously pigmented or rarely red due to hemoglobin in the immature stages of few aquatic and endoparasitic flies, e.g., chironomid larvae. Hemolymph performs the function of both blood and lymph. It is not involved in gas transporting function, respiration. Hemolymph contains a fluid portion called plasma and cellular fractions called hemocytes. 1. Plasma. Plasma is an aqueous solution of inorganic ions, lipids, sugars mainly trehalose, amino acids, proteins, organic acids and other compounds. pH is usually acidic, 6.7. Density is 1.01 to 1.06. Water content is 84 to 92%. Inorganic ions present are Na, in predators and parasites, Mg, and K in phytophagous insects. Carbohydrate is in the form of trehalose sugar. Major proteins are lipoproteins, glycoproteins and enzymes. Lipids in form of fat particles are lipoproteins. Higher concentration of amino acids leads to a condition called aminoacidemia, which affects the osmosis process. In high-altitude insects, glycerol is present, which acts as an anti-freezing compound. Nitrogenous waste is present in the form of uric acid. 2. Hemocytes. The blood cells or hemocytes are of several types and all are nucleate. Different types of hemocytes are as follows. A. Prohemocyte, smallest of all cells with largest nucleus. B. Plasmatocyte phagocyte, aids in phagocytosis. C. Granular hemocyte, contains large number of cytoplasmic inclusions. D. Spherule cell, cytoplasmic inclusions obscure the nucleus. E. Cystocyte coagulocyte, role in blood coagulation and plasma precipitation. F. Enocytoids, large cells with eccentric nucleus. G. Adipohemocytes, round or ovoid with distinct fat droplets. H. Podocyte, large flattened cells with number of protoplasmic projections. I. Vermiform cells, rare type, long thread-like. Functions of hemolymph. 1. Lubricant. Hemolymph keeps the internal cells moist and the movement of internal organs is also made easy. 2. Hydraulic medium. Hydrostatic pressure developed due to blood pumping is useful in the following processes. A. Ecdysis, molting. B. Wing expansion in adults. C. Eclosion in Diptera, adult emergence from the puparium using telinum. D. Aversion of penis in male insects. E. Aversion of osmeteria in populionid larvae. F. Aversion of mask in naiad of dragonfly. G. Maintenance of body shape in soft-bodied caterpillars. 3. Transport and storage. Digested nutrients, hormones and gases, chironomid larvae, were transported with the help of hemolymph. It also removes the waste materials to the excretory organs. Water and raw materials required for histogenesis is stored in hemolymph. 4. Protection. It helps in phagocytosis, encapsulation, detoxification, coagulation, and wound healing. Non-cellular component like lysozymes also kill the invading bacteria. 5. Heat transfer. Hemolymph through its movement in the circulatory system regulate the body heat thermoregulation. 6. Maintenance of osmotic pressure. Ions, amino acids and organic acids present in the hemolymph helps in maintaining osmotic pressure required for normal physiological functions. 7. Reflex bleeding. Exudation of hemolymph through slit, pore etc. Repels natural enemies, e.g., aphids. 8. Metabolic medium. Hemolymph serves as a medium for ongoing metabolic reactions. Trehalose is converted into glucose. Lecture 12. Anatomy. Nervous system. Nervous system. To view the ventral nerve cord, examine the ventral region of the roach's body cavity, or specimen you performed the dorsal dissection on, for something that resembles a railroad track running from the head posteriorly to the abdominal region. The railroad track is made up of 2 nerve cords, connectives, that run longitudinally with a series of node-like ganglia. The anterior most region of the ventral nerve cord is called the subesophageal ganglion. Just dorsal to that structure is the insect, brain, or supraesophageal ganglion. The basic component